In today's lecture, we will talk about post-translational modifications, structural proteomics, role of bioinformatics, challenges and future direction. So, let us start with PTMs. Post-translational modifications are vital cellular control mechanisms known as cellular switches that affect protein properties such as protein folding, conformation, activity and functions. As a result, they play very important role in various diseases. The protein complexity arises due to gene splicing and post-translational modification. Once the protein is synthesized by the ribosome from its corresponding mRNA in the cytosol, many proteins get directed towards the endoplasmic reticulum for further modification. Certain N-C terminal sequences are often cleaved in the endoplasmic reticulum, after which they are modified by various enzymes at specific amino acid residues. These modified proteins then undergo proper folding to give functional proteins. Due to these modifications, the number of proteins are 3 order of magnitude higher than the total number of genes encoded in genome. There are several types of post-translational modifications that can take place at different amino acid residues. The most commonly observed PTMs include phosphorylation, glycosylation, methylation as well as hydroxylation and acylation. Many of these modifications, particularly phosphorylation, serves as regular mechanism for the protein action. PTMs generate tremendous diversity and are extremely important. Many documented effects of post-translational modification include change in enzymatic activity, ability to interact with other proteins, subcellular localization and targeted degradation. The final structure of functional proteins most often does not correlate directly with the corresponding gene sequence. This is because of PTMs that occur at various amino acid residues in the protein, which cause changes in interactions between the amino acid side chains, thereby modifying the protein structure. It further increases the complexity of the proteome as compared to the genome. The protein phosphorylation acts as a switch to turn on or turn off the protein activity and governs wide range of polypeptides from transcription factors, enzymes to cell surface receptors. The reversible phosphorylation of proteins catalyzed by kinases and phosphatases regulates important cellular functions. Phosphorylation of amino acid residues is carried out by a class of enzymes known as kinases that most commonly modify side chains of amino acids containing a hydroxyl group. Phosphorylation requires the presence of a phosphate donor molecule such as ATP, GTP or other phosphorylated substrates. Serine is the most commonly phosphorylated residue, followed by threonine and tyrosine. The removal of phosphate groups is carried out by phosphatase enzyme and it forms one of the most important mechanisms for protein regulation. Glycosylation involves linking saccharides to proteins in presence of glycosyl transferases enzymes, giving rise to a glycoprotein. Glycosylation play vital role in various biological functions such as antigenicity of immunological molecules, cell division, protein targeting, stability and interactions. The aberrant glycosylation forms result into various human congenital disorders. Depending on the linkage between the amino acid and the sugar moiety, there are 4 types of glycosylation: N-linked glycosylation, O-linked glycosylation, C-mannosylation and glycophosphatidyl'inositol anchored GPI attachments. Glycosylation involves the enzymatic addition of saccharide molecules to amino acid side chains. This can be of 2 types: N-linked glycosylation, which links sugar residues to the amide group of asparagine, and O-linked glycosylation, which links the sugar moieties to the hydroxyl group of serine or threonine. The glycosyl transferase enzymes catalyze these reactions. Sugar residues that are attached most commonly include galactose, mannose, glucose and acetyl glucosamine, etc. Protein mixture is digested with proteolytic enzyme such as trypsin, and resultant peptides can be analyzed by MALDI-TOF or LC-MS. The top-down mass spectrometry involves analysis of intact protein using high-resolution mass spectrometry techniques. High-resolution MS platforms such as FTICR-MS, ORBITRAP-MS, with PTM-friendly dissociation techniques such as electron capture dissociation and electron transfer dissociation (ETD) are commonly used. PTMs can be detected by means of mass spectrometry due to the unique fragmentation patterns of phosphorylated serine and threonine residues. The modified protein of interest is digested into smaller peptide fragments using trypsin. This digest is then mixed with a suitable organic matrix such as alpha cyano 4 hydroxy cinnamic acid, cinnappinic acid, etc., and then it is spotted onto MALDI plate. The target plate containing a spotted matrix and analyte is placed in a vacuum chamber with high voltage and short laser pulses are applied. The laser energy gets absorbed by the matrix and is transferred to the analyte molecules, which undergo rapid sublimation resulting in gas phase ions. These ions are accelerated and travel through the flight tube at different rates. The lighter ions move rapidly and reach the detector first, while the heavier ions migrate slowly. The ions are resolved and detected on the basis of their m by z ratio, and a mass spectrum is generated. Identification of post-translational modification by MS largely lies in interpretation of results. comparison of list of the observed peptide masses from the spectrum generated with expected peptide masses enables the identification of those peptide fragments that contain any post-translational modification due to added mass of a modifying group. In this hypothetical example, two peptide fragments are found to have different m by z values, differing by 80 Daltons and 160 Daltons. It is known that the added mass of a phosphate group causes an increase in m by z of 80 Daltons. Therefore, this principle of mass difference enables the detection of modified fragments. Affinity-based enrichment, immunopurification and metal affinity chromatography are commonly employed for purification of proteins containing specific PTMs. Immobilized metal affinity chromatography (IMAC) and metal oxide affinity resin such as titanium dioxide, Fe3O4 are also commonly used for the enrichment of phosphoproteins. A protein phosphorylation experiment is shown here. The complex protein sample is loaded onto a miniaturized affinity column which interacts specifically with proteins having the post-translational modification of interest. The IMAC chromatography columns containing ions such as zinc, iron, titanium dioxide specifically chelate the phosphorylated proteins. The unwanted proteins are removed by washing the column with a suitable buffered solution, after which the phosphorylated protein of interest can be eluted out by modifying the buffered solution. The protein purified by liquid chromatography is then subjected to triptych digestion, followed by analysis using tandem mass spectrometry. Here I have demonstrated the use of multi-TOF MS for resolution of the generated ion fragments. Separation is based on the flight time of the ions and greater resolution is achieved due to the presence of two mass analyzers. The peptide ion spectrum generated is analyzed by comparing it with the expected spectrum, thereby allowing determination of modified peptides having different m by z values. The metabolic labeling methods such as SILAC is used for label based quantitation of PTMs. However, this strategy can only be used for the living cells. Other chemical labeling methods such as ITRAC is also used for PTM analysis. To predict the structure and biological function of an uncharacterized protein, computational methods rely on structural homology of unknown protein from proteins with known structure and biological function. By relying on such methods for structure function correlations, it is possible to predict biochemical function of uncharacterized proteins based on structural homology to another protein with a known function. Recent advancement in proteomics and other omic technologies allow large-scale analysis of biological samples and generate an unprecedented amount of digital data. In different modules, we have discussed different bioinformatic tools and software for analyzing proteome and system level investigation using two-dimensional electrophoresis, mass spectrometry, microarrays and surface plasmon resonance. Computational challenges associated with proteomic studies have recently emerged as some of the most critical and limiting factors in this rapidly evolving discipline. Bioinformatic tools have been widely used for protein sequence analysis. It is also used for protein motif detection and epitope prediction, active site determination, determining transmembrane domains, as well as identification of DNA binding residues. Database designing is done at various levels such as physical, logical and conceptual. At the physical level, the purpose of the database is defined, which is in accordance with the proposed usage. At the logical level, the tables, attribute of the tables and data types are defined. At the view level, the views and appearance of the databases are defined. A typical biological database can be characterized by its type and its tool. The type defines the category of data that it includes, such as sequence, domains or structure. This implies that the particular database its most prominent feature include either sequences, domains or structure, and it is particularly used for their analysis. The analysis tool defines the platforms that the site will provide for gaining an insight into the protein data. For extracting the protein information from a database, users can give a variety of input terms. These can be unique ID, molecular name, amino acid sequence, keyword, literature, gene, taxonomy, etc. Once the user submits the query, the output can be of multiple formats. The generalized information that users can obtain from protein databases is general description of the protein molecule. The generalized information that users can obtain from protein databases is general description of the protein molecule, annotation of the protein, name and description of the gene that transcribes them, ID of the same protein in other relevant databases, details of the experiment conducted for characterizing proteins, details of proteins secondary structures, details of the organism which was used as a source for obtaining the protein and citations of research conducted. Database analysis tools. The different kinds of analysis can be conducted on a given protein sequence. The query can be protein name, sequence or any other identifier of the protein. Various kinds of results output can be obtained. Identity of protein from sequence, identify physicochemical properties, molecular weight, isoelectric point, sequence tag information. Similarly, search algorithms such as versions of BLAST, FASTA and multiple sequence alignment. Finding conserved and variable domains in the protein to study its evolutionary relationship with other proteins, molecular modeling and visualization tool. Secondary and tertiary structure prediction and structural analysis, biological text analysis such as biomedical acronyms, gene protein synonyms, etc. Database mining in proteomics and visualization tools. Collective improvement in any research field can be accelerated by sharing scientific data among different research groups across the world, seeing as it allows other researchers to access, validate and reanalyze one's finding and correlate the results with their own observations. Data management is critical when using high throughput proteomic techniques. Several internet databases have been established to collect the proteomic data sets. Data Enabled Life Sciences Alliance (DELSA) is a timely and important initiative to create a common data bank where on one hand we can access the huge data set generated by various research groups. On the other hand, we can also deposit our data sets which may be useful for a wide range of researchers working in similar fields. At present, the broad field of DELSA encompasses biological sciences, ecology, environmental sciences, evolution, genomics and proteomics, computer sciences, cyber infrastructure, management, health sciences and policies for global distribution. MS-based proteomics encounters the following biological problems while analysis of huge number of proteins: fragile nature of proteins, substantial losses occurring during the sample collection and processing steps, presence of multiple isoforms of single protein, the wide dynamic range of protein concentration in biological fluids, presence of high abundance proteins masking low abundant marker proteins. Additionally, the technological limitations associated with most of the commonly used MS-based approaches include typical dynamic range of only 10 to the power 2 to 10 to the power 4, inadequate coverage of whole proteome unless sample is fractionated extensively, low throughput and issues of robustness and cost, overfitting the data, machine fluctuation, instrument noise and contaminants in spectrum and lack of standard procedure for analysis and interpretation of MS and MS-MS spectrum. To overcome these technological challenges, different novel and amalgamated approaches have emerged in last few years. The most promising advancements include large scale quantitative proteomics, culture derived isotope tags and superciliate based technologies. Multiplexing, tandem mass tags, TMTs and high-track 8-plexing, quantitative accuracy, label-free LC-MS-MS, low sample consumption and large-scale analysis chip-based and nano LC-MS, sensitive quantitation of proteins within complex mixtures, biomarker discovery, multiple reaction monitoring MRM-MS, large scale biomarker discovery, etc. Impending future of this promising research area will highly depend on the collaborative initiatives at global level and establishment of effective data repositories accessible to the proteomics researchers across the world. In 2010, Human Proteome Organization (HUPO) has launched a Global Human Proteome Project (HPP). This project is designed to map the entire human proteins encoded by the genome. Let us now discuss some of the targeted focused initiatives. The Human Liver Proteome Project. This is the first initiative of Human Proteome Project for human organ, tissues with an intention of generation of comprehensive protein atlas of the liver and international liver tissue network, collection and distribution of normal liver samples and validation of new discoveries. Plasma Proteome Project. Analysis of the protein constituents of human plasma and serum. Human Brain Proteome Project (BPP) focuses on the revelation of the brain related proteomic alterations, focusing on understanding neurodegenerative diseases, aging and identification of prognostic and diagnostic biomarkers. Human Kidney and Urine Proteome Project aims to understand kidney functions, mechanism of chronic kidney diseases at a protein level and discover biomarkers and target molecules for new therapeutics of kidney diseases. Over the last decade, proteomics research is progressing in different regions of India with a considerable interest. India is playing an increasingly significant role in global genomics and proteomics research and development, as it is evident from research publications and patents. Indian government is also supporting the basic and applied proteomics as well as other omics based research, and multiple national and international funding agencies are providing investment on existing and new research projects. Considering the space of emerging proteome level research, it can be anticipated that in coming future, some amicable solutions for the existing limitations associated with the burgeoning field of proteomics will come forward through worldwide research initiatives, and this discipline will become more robust, sensitive, reliable, rapid, cost effective and user friendly for resolving real life biological problems. Hope this course has given you foundation for proteomics concepts and enthused you for research in proteomics area. Thank you.